and on behalf of the participants of this AICTE sponsored ATAL FDP program. Welcome you, sir, Professor Pawan Dhar.
Thank you very much, sir. So nice of you for your kind introduction, Professor Balakrishnan, and thank you for inviting me here on this very important platform. I'm so delighted that when I returned from Japan back in 2009, and then joined Kerala University in 2010 to start the center on synthetic biology. At that time, we didn't know who are the people who are interested in this area. And so we organized this Biodesign India meeting, October 2010. And then the synthetic biology meeting community had started crystallizing. So I'm happy that a couple of speakers in this series, they are from the original group that talked about synthetic biology in this country nearly a decade back. And Professor Guhan Jairaman is one very eminent scientist who came and he gave a talk there. And also Kapila, who is giving a talk towards the end, Dr. Kapila Kumari. So she also trained with us, and it's wonderful to see that there is a lot of interest. So I'll share my slides. So the whole thing is to first understand what is synthetic biology, and why are we talking of synthetic biology, in this current context, and is it another name, or is there some really good science behind this, which people are, connecting with? Because my introduction to synthetic biology also happened by chance. I was in the US for some reasons, and there was in June 2004, there was a first synthetic biology meeting at MIT in Boston. And I happened to attend that, not because of the meeting, because at that time, nobody knew what is synthetic biology. This name was not there anywhere in the community. I just want to see MIT, and connect with some people there in Boston University and some of my friends and previous colleagues. And I happened to be in this place. And this was a 3-day meeting, which totally opened a new perspective. And at that time, I was working in Singapore and heading the systems biology group. So after these 3 days of very, very nice discussion, I came back and then I decided to leave systems biology and get into synthetic biology full time. And this is what happened. It was not programmed. It was just by chance. And so whatever introduction I'm going to give you, it will be originating from this meeting, the MIT meeting, wherein the need for synthetic biology was very clearly highlighted, and the justification for the new name was expressed. So as you know, the way science has happened is by reductionism. So which means that there is an organism and you want to understand how this organism is working, and to explain the behavior of an organism, you dissect the organism to see what is it composed of, what are the systems it is composed of. The moment you reach these details and you get some nice pictures and nice description, you want to know a little more, because this itself doesn't tell you everything. So you want to go a little deeper. And so after this, the next level that people went for was finding the description of the organs. How are they connected? Now, from systems, you're going to the organs. How are these organs connected? And once you know the input-output values of these organs, you also want to know what is the origin of these input-output values. Then you go back to the down deeper into the cell level. And early on, when the cells were, the staining technique was not so sophisticated, cells were looking like innocent structures. But as the microscopic technologies and staining technology improved, many orders of magnitude in terms of the resolution of the object, more and more details started coming up. And so the cell, which was not giving much details other than the nucleus, now a lot of things started appearing. And one of the molecules that took the center stage back in early 40s and 50s was chromosome. And this chromosome was found to be made of DNA. And the DNA with proteins together, it was found that they both combine, and then DNA winds itself 7,000 to 10,000 times in order to make chromosome, and also unwinds itself. So the DNA early on, back in mid-50s, was considered as the main key molecule by the community. And so it took a lot of headlines for a couple of decades. But over time, when more and more details were uncovered at the RNA level and the protein level, and then not only the mRNA, but also the microRNA, the regulatory RNA, and people started getting a feel that DNA is only a database. So this is how the science has evolved. And when people went a little more deeper into this, so what is the next level after DNA is the molecules. So now from biology, we're entering the domain of chemistry. Because it happens in biological setting, we call it biochemistry. But beyond molecules, when questions started appearing at the interface of these molecules, that what are these molecules doing, how are they organized, then people entered the space of fundamental physics. Now, when you go deeper into this, the trouble is that 99.9% of atom is space. Now, you cannot have a microscope to stain the space or to observe the space. We don't have the technology as yet. We cannot use GFP to, illuminate the space. So now we'll reach the limits of resolution. And of course, now this becomes the domain of physicists who go to the subatomic world and find out there are some more particles which are fundamentally involved. So the journey of reductionism, which started from the phenotypic observation, if you go deep enough, then you're crossing the boundary of chemistry and then physics. So the point is, the whole process itself is called reductionism. You see a particular object, and then you try to see what is the object made of. And when you have a lot of data, let's say 90s, early to mid 90s, there was efforts to do the omics, genomics, and then transcriptomics and proteomics and so on. It's still going on. As a result of these omics efforts, a lot of parts were uncovered. So a cell which was looking, with less description, with the less resolution of the microscope, suddenly has got thousands and thousands and thousands of parts. And now these parts manually cannot be listed, and they cannot be tabulated, or they cannot be even understood. So now there's a need to capture this data in the computer. So which means that reducing the system to a set of parts is one approach in science. Capturing these details and transferring the data to computer is another approach of science. Okay, so these are the two fundamental approaches of doing science. The third approach came in when in 2004, scientists, in fact, they were engineers at MIT, who proposed the construction. So they said, now that we know the reductionist view of life, we also are able to integrate the data and build databases. Can we construct the system from scratch? Can we chemically synthesize a genome? Can we chemically synthesize a chromosome? Can we chemically synthesize a mitochondria, Golgi body, or the whole cell? That was the question which was asked early on. And that is the one which led to this new approach of building things in biology. So the first science of reducing the system to a bare-bone set of components, we popularly call that as molecular biology and biochemistry. The other science, which captures the data and then transfers that to database for deeper analysis, is called as bioinformatics and systems biology. Bioinformatics, if you're dealing with one part, systems biology, if it's a bioinformatics of the network, the whole cell bioinformatics, we call it as a systems biology. And the third, which is about constructing things from scratch, given a set of genes, can we make a genome, or can we construct the cell? So such kind of efforts are called synthetic biology. So I hope that you got the idea of what is synthetic biology and why it was proposed as a new kind of science, why it didn't fit into any other science which was available at that time. So I hope people have a fair sense of what is synthetic biology. So the question again is, can biologists think like engineers? Yes, because there are many similarities between engineering and biology. Both are robust, linear and non-linear, with multitasking events. Both are based on analog and digital, and robustness to fault or the error is found in serial and parallel processes running in both the systems. So a cell is a massively parallel and massively interactive system. So therefore, because of the similarity of the engineering and biological systems, you have motivation for this engineering approach to design biological systems. However, there are also differences because in engineering, you handle anchored components. You handle components that are fixed to a certain surface, and you call it motherboard. And you attach these transmitters or transistors, and the resistance and the capacitors and the wire that connects them. You are all fixing them to a certain surface. While as in biological systems, you have RNA and protein molecules that are moving all the time. They're not anchored to something. So imagine if you have to make an electrical wire equivalent in biology, biological wire or a DNA wire. Then how do we do it? A gene expression wire, which means that it's a kind of a waterfall model wherein one process leads to another process to another process. So you're talking of floating components. In engineering, it's predominantly digital, and biology is predominantly analog. Digital means that you switch on the switch, and it is either on or off when you switch on the tube light, for example. But when you're dealing with the fan regulator, you see that this is 0, 1, 2, 3, 4, 5. It can change from the lower value to the higher value. That means a graded response is available when you talk of the fan regulator, and that is an analog system. In digital systems, in engineering systems, you always have unidirectional flow of information at one particular step. So in that step, the information will go either forward or reverse. It will not go both sides, both ways. But in biological systems, you have reversible flow. Say, for example, at the when the substrate and enzyme combine to make a product, you have a transitory structure called enzyme substrate complex. And where this enzyme substrate complex decomposes to form substrate at a certain rate constant. So at one particular step, you have a reversible flow. So how can we handle that when you're designing new systems? Because part of the information comes back. Engineers are lucky because they use the laws, rules, and principles, generated by physicists. But biologists have to settle down with Mendelian laws. If you call them laws, some people like to call them as ratios rather than laws. In any case, these laws or ratios, they are zoom in, zoom out type. So you zoom in from phenotype to genotype, and you zoom out from genotype to phenotype. But the layers that are within this they're not considered, and the laws were not designed for that purpose in any case. So we cannot use them. Noise is filtered in engineering systems, but noise is used. When you say noise, I'm talking of gene expression noise, the fluctuation that goes up and down. Likewise, there are other dissimilarities. So here you see a sample of the biological expression noise, the gene expression level, and it's normal. So if you say if you ask somebody what is the signal intensity in an electrical wire, maybe they will point to the ampere or voltage, and they say, okay, this is a fixed value. But that is not the case with the biological systems, because the expression always goes up and down. It's stochastic expression, and therefore it becomes harder to create systems using biological infrastructure. In case of electronic circuits or engineering systems, it is pretty straightforward to place the information nodes and to decide in what way the information is going to travel, at what point it's going to travel, how long and how far it's going to travel, in what direction, magnitude, and all that it's going to travel. But when it comes to biological systems, creating an equivalent of engineering circuit or electrical circuit would mean that mRNA has to be formed and protein has to be formed. If protein is able to talk to the next DNA sequence, then the next event is switched on. Now, this is a low resolution of biological circuit. If you go at the high resolution, the resolution at which it actually exists, you will notice that it is not the making of mRNA only that decides the next event or the making of the protein. It is also how far or how long the mRNA or protein is surviving. So if mRNA is degraded, normally mRNA is degraded at a very, very high level, and a very tiny portion is taken to the level of making proteins. Now, if protein survives long enough and also is able to find the next promoter sequence, only then you will have a possibility of making a biological circuit. So what I'm trying to indicate here is that it is quite difficult to create biological circuits as against engineering circuits, and because of the reasons that are very clear now. So to design well-behaved systems, one needs standards and rules of compositions. At least you need a standard. A standard is a quantity that is always fixed. The rules of composition means that in what order do you combine the components to make a certain system. So, for example, one of the examples of standard could be when we see the railway tracks, they are laid with a certain fixed distance between the track. This cannot be more, this cannot be less. It has to be absolutely same. And a certain standard is fixed that because the railway coaches are made based on this distance, therefore it is very important that we keep this distance fixed. These are called standards. Now, standard does not mean that this is the best distance, because you have a broad gauge, you have a meter gauge, you have a narrow gauge. There are many standards which people have followed. But the question is that what do you keep as a fixed quantity? That is very important. Now, it can be 5 feet 6 inch. It could have been 5 feet 10 inch. It could have been 5 feet 11 inch. It doesn't matter. But once it is fixed, it's fixed. This is what we know as a standard. Rules of composition. Now, let us say you have to assemble this watch. Unless you know in what order you are supposed to combine the parts, even if every component maps to a certain standard, you cannot make a watch, because you need to know in what order do I need to place the components on the top of each other to make a watch. So two things are always needed. One is the standards and the other one is the rules. So the grand challenge definitely in biological engineering community is to create brand new cells and multicellular consortia of cells based on engineering standards. So now the question is, how do you build the standards in biology? Eventually, it's all trial and error. It is also that, say, for example, if you say that the way DNA is isolated in my lab, everybody should isolate DNA in the same way. So this is a global standard of DNA isolation. People will not agree, because they are using different strain, they are using different cultural conditions, they are using different DNA compositions, or sometimes DNA is within the nucleus and sometimes it's out of the nucleus. So you have different varieties of methodologies. So the standard would mean that what is fixed is fixed. It cannot change. So for example, in this equation, you see a lot of variables. You see coefficient, you see operator, you see constant. Here, in terms of the standard, these are the 4 spots which are fixed in the equation. These are not changing. The rest of it can change. So this is what I'm trying to say is an example, an imperfect example. It's not the perfect example, it's an imperfect example of how the biological standards would eventually evolve and look like. For example, in E. coli, there are certain constants which always, or not always, but more or less stay the same way. And as you can see this in this picture, we make use of these numbers in the models, in the computational models. But if we have to create a brand new E. coli, chemically synthesize it, and pack it with a lot of components, then we would need to have a reference point of this kind. So the IEEE standards, which are very common in engineering community, they do not exist in biology because organisms were not created to solve human problems. So therefore, it is good to use engineering as a philosophy instead of engineering tools, which means that if you copy and paste engineering textbook equations, it will not matter because these are two different domains. The approach is understandable, but the path has to be unique. Absolute engineering solutions for biological problems do not exist. Probably engineers never faced a problem as complex as this. This is not to downgrade the efforts of engineers who have been able to reach all the way up to moon and beyond, which is a mega Herculean task. However, what we are trying to say here is that there are certain features in biological systems which are not found in engineering systems because of the uniqueness of these features. Biological decisions are taken based on probabilities. They're taken based on gradients, especially if you look at the cell, the development of an organism. You have a concentration gradient, and then that decides a lot of things during the development. And you have one special feature in biological systems called emergent phenomena. Emergence means that 2 plus 2 is never equal to 4. It can be 5, it can be 3, it can be some value in between, but it is something which is an unexpected behavior from an organism. Because when the components talk to each other, remember this is analog system, nonlinear system, and when they come together, they generate a behavior that could not have been predicted by using the component description alone. So therefore, when you knock in a gene in a cell, you realize that this knocking in, what kind of effect it will create in the cell, nobody can predict. You knock out a gene, no one can predict what kind of effect it can create. So this is what I'm saying, emergence. We're just waiting and watching for organisms to react and then learn from there. So what's the solution? Well, it is not a solution as such, but it's a practical approach, the way forward that traditionally we have used. We have used a top-down approach to decompose the system one part at a time. The new focus is to compose the system one part at a time with the hope to do controlled construction. Now, this is very important because whatever you construct has to be under a controlled environment, and the outcome has to be absolutely predetermined. So that terminologies like truth table have come into the vocabulary of biologists after this meeting, 2004. What it indicates is that a table, which is a quantitative truth table, wherein the inducer or repressor concentration is put on one side and the protein concentration on the other side, thereby indicating a linear correlation between the concentration of inducer and the corresponding outcome. So a box in this table would indicate the state of the system at that point in time. Now, using this truth table, engineers have constructed a large, complex, machines and circuits. The hope was that we can do the same thing. But this project was started at MIT by a couple of students, and they took up this as a PhD project, and they found it very difficult to convert an analog circuit or an analog expression of the genes into a digital expression. So the whole point was that the truth table and the whole logic gates, that will work beautifully once you have a digital expression of the genes as against the analog. Analog means that it's a wavy expression going up and down, but digital expression means that it goes straight and then suddenly it goes up and then takes another value, which is again a stable value. So you have a ground level of expression, and then you suddenly go for a higher value, and that value gets stabilized. So this would be a a step function or a digital expression, which didn't happen because of the nature of the system itself. So to convert an analog into a digital expression itself, it's very, very difficult. And even if people have achieved somewhere, the expression gets diluted with the cell division, or the expression has to be maintained in a great number of constraints. So it's not something which can be scaled up or merged with the other genes. So concepts like logic gates have also found their entry into biological community. So you have a NOT gate wherein, for example, you have a lac operon wherein the repressor is on and the output is off, and the repressor is off and the output is on. So you have a classic inverter switch. Or when you see that the activator and inducer together are needed for the product output, then you think of this as a something of a AND gate. Well, the gate representation is definitely worked out. There are different kinds of gates, and people have figured out, and also combine different gates to make it more complex. But at the same time, we need to be sure that this is not the kind of rocket we are trying to build and launch in biological community, because it will always come with some surprises. So it's very difficult to maintain that accuracy as the complex grows further. So synthetic biology is the rational design and controlled construction of biological components leading to applications. Back in June 2004, when the term synthetic biology was announced, there was a debate after the meeting, and it went on for more than a year, wherein people wanted to know why synthetic biology. And some people didn't like it because it gave a flavor of chemical biology. So they suggested many terminologies, new names were proposed. And one of the names was constructive biology. And so people said that if synthetic biology is constructive biology, or construction biology is the rest of the biology destructive. So this was not accepted. Then people said, let's call it biological technology. And again, there was objection that if biological technology is the new name, then what is biotechnology? Then some people said, let's call it biosystems engineering. And then people have different ways of, naming this new approach. If biosystems engineering is the way to go forward, then how about the components that make the system? So we have to engineer the components also, not only the systems. So finally, people settled with this terminology called biological engineering. And so it was distinct from bioengineering. Well, it was very similar, but at the same time distinct, because bioengineering is more of biomedical engineering, wherein you have devices to support a patient. However, here the idea is to engineer the system ground up and leading to applications. So if you come across papers or journals on biological engineering, it would mean the same thing as synthetic biology. So synthetic biology and biological engineering means exactly the same thing. It's again a matter of preference. So back in June 2004, when the first meeting took place and the field was formally announced, there was one speaker by name Ron Weiss from Princeton University. So after this meeting, I approached him and I asked him, I said, well, the field has been announced, but we don't have a journal in the field. We have a responsibility to create a journal. So how about we team up together and we launch a new journal? So he agreed, and we launched this new journal called Systems in Synthetic Biology with Springer. Earlier it was with Biomed Central, then we moved in 6 months' time to Springer. And then we ran this journal for nearly 7 years. And it had to be closed because by then a lot of other organizations came up and there was fierce competition and the space is very narrow. And then established journals like Nature and Science and Cell and other journals, they also started synthetic biology sections in the journal. So it was pretty hard to compete. And ACS, for example, started synthetic biology journal. So we said it's time to say goodbye and go back to your labs. So biological engineering journal, Journal of Biological Engineering, also started something around 2009, 2010, and has been doing pretty well ever since. So there are many opportunities for people who want to get involved in the field, and one of the opportunities is the annual IGM competition that is held in MIT, October last week to November first week. That's the usual time. And several years ago, I proposed this to DBT that we should have our own Indian version of the IGM, wherein we may not have the infrastructure, but at least some provision for people to basically get the funding and present their work at MIT. So a new initiative called IBEC was launched. IBEC would be Indian Biological Engineering Competition. And as a result of this IBEC, we propose that 10 lakh should be given to each team, a maximum of 10 lakh should be given to each team. And depending on their own resources, of course, that's not enough. But depending on their own resources and ability to get funding from other sources, they should be able to present their work at MIT. So this has been very successful. Every year, we award at least 5 teams. We award this funding, and a number of teams from Chennai, from Bangalore, from Pune, from Delhi, they keep on applying, and we encourage people who have never competed in this meeting. So I would really encourage, and I will invite all of you, if you're interested in this competition, please feel free to apply, and the applications will be open very soon, maybe in January. There's Dr. Sangeeta Kasturi, who is responsible for this. And I would really encourage people. So don't think that we are from a place which is not very well known, and so whether we'll get funding or not. We have even turned down proposals from established institutes because we want to encourage newbies and newcomers in this area. And you will see that Dr. Kapila, who is going to present her work, she comes from a university close to in Haryana, and so we have been supporting a lot of people and from places which are not so established in this area. They may be well known in their other areas, but this area may be new. So please do feel free to apply, and I'll be happy to answer questions regarding that. We also wanted to create a scale up this IGEM in the sense that we want to create the IBEC on the lines of IGEM, but at the moment we don't have funding because this needs committed funding. This needs very strong support because if you have to distribute the clones, if you have to create a standard part inventory in our own country, it would need a dedicated effort, a dedicated team, and a assured funding. So DP is open to this. If you have any suggestions or you want to take the initiative, you're most welcome to write to me, and we can take it forward. So as I was saying that, this is the Indian Biological Engineering Competition, and applications are invited. So please feel free to apply. And also there is a Global Alliance of Biofoundries that has been created, and it is led by Paul Fremont from Cambridge. So you can see that in India, there is no player at the moment, and there are players from East and West. So in case you happen to have some interest in this area, and one of the things I would like to mention here is that it's important to, when you get excited about some new field, it's important to take responsibility and make sure that you push the boundaries further. So if you're interested in this or the IGM, please feel free to write to me. and you have this website called biofoundries.org. And as you can clearly see that it's time for somebody in India to take initiatives, and you can write to them directly, and you can read more about it. Likewise, in US, very early on, SynBio, which was Synthetic Biology Engineering Research organization or cluster, that was proposed, and a lot of academic organizations and company joined hands together to create this organization. But it later on changed to EBRC, and now it is under a new umbrella. And they also offer internship programs. And so please go through these possibilities and see what is interesting in your context. People have used engineering approaches to like refactoring is one of the very popular term. That means that you're separating. If there's an overlapping, you're separating. So Drivendi at MIT, who actually started this field, so in his lab, they said that if we separate out the overlapping genes, the overlapping reading frames, and then put them under control of an independent promoter, what will happen? So they just started separating this overlapping, reading frames, and they found that as they refactor the genome, the colony size is shrinking, and the properties are also getting changed. Which means when we say that it's a nested expression or an overlapping, it is not something which is random, which is something which needs to be corrected. This is an optimal way that organism has found its way to survive. There are people who are now thinking of moving beyond the nucleosides, the nucleotides that have been evolutionarily available, and create brand new DNA. Well, there are of course many aspects highlighted here at the biosafety level, but so far we do not have a fully functional organism with a non-natural nucleosides, but people are working towards that. One of the landmark publications came from Professor Chandra's lab, and he was the PhD guide of Dr. Aniluru, and the whole idea was that can we take yeast chromosome, split it into smaller portions, chemically synthesize each part, stitch that part in yeast, and then reboot a yeast with a brand new chromosome, which is redesigned. Imagine this is taking an existing chromosome and slicing it in the computer in small regions, taking away those regions which are not necessary for the chromosome. Evolutionarily, there are some, some, so to say, junk regions. They may not be totally junk, but they may be there for evolutionary reasons. And then redesigning the chromosome and making it lighter. Can we do this? So his work, he spent nearly 5 years on this, and his work showed that it is possible to chemically synthesize chromosome, and a fully functional chromosome was chemically synthesized. So it was such an important paper in science that Craig Venter wrote an opinion on this. So this was one of the landmarks development. Also, the long DNA synthesis has been has come up as an alternative to the recombinant DNA technology. Like you combine DNA sequences into different patterns, and you reboot the cell with a recombinant vector. The question is, can we chemically synthesize a recombinant DNA vector? Chemically synthesize it. So people are trying to design the chromosome or the vector. I'm sorry. They're designing the vector in the computer, and then the output of this vector is a series of ATGCs. It's a text file they send to the chemical synthesis company, and the company synthesizes the vector and delivers the recombinant vector in a matter of 3 weeks or 4 weeks, depending on the complexity on the size of the vector. So something that used to take a year or more, that can be done within a month. This is how fast the DNA synthesis technology has proceeded. And also the genome editing has taken a center stage, and now we have a Nobel Prize coming up in that area. And the world's first synthetic cell was synthesized by this group. It took them 10 years to do this experiment and a lot of breakthrough technologies. But finally, they rebooted a cell with a brand new genome. Likewise, in industry sector, there have been many examples of, let's say, making isoprene, and that's using Goodyear company to make rubber tires, and bioacrylic, made from sugar by the yeast cells. And this, as you can see, is a lot cheaper and also environment friendly. Likewise, you have metabolics company making biodegradable plastic from the corn. So the question is, do we have enough parts? Now, we have tried to handle this question that we are talking about a lot of genes made in every organism. Now, we also know that, say, for example, in human genome, there are 1.5% is supposed to be protein coding, and 90% or a little more is supposed to be RNA coding. Now, the question is that if it is 1.5% is protein coding, and can we convert rest of the genome into protein coding parts? So, therefore, we took up this question, and in our lab, we have a very simple mission of making our own genes. Traditionally, by converting genes into junk and garbage, we have learned biology. Now, the question is that can we reclaim this, so to say, junk DNA and do something useful? Can we make brand new genes from the so-called junk DNA? When I'm talking about brand new genes and proteins, I'm talking of sequences that are neither expressing RNA nor expressing proteins. So can we use this? I'm getting some background, so if everyone can switch off their audio, please, that would be a little more comfortable. The point is that when we have three kinds of genome sequences, one is coding for protein, the other is coding for RNA, and the third is not coding for anything, which is called more of a dark matter. Can we make some brand new peptides and proteins from there? That was the question which we asked. So this is also coming from the observation that nature placed start and stop signals at certain specific spots in the genome. What made nature decide that the expression should start and end there? And if it was randomly done, then there is scope of more information coming out. So that means in the same sequence, you can make more genes because it's all information. Is it possible? Can we make genes from intergenic sequences, brand new genes, and brand new functional proteins from intergenic sequences? That is what we asked. And we took, as you can see, the red line, the ICA 1, ICA 2, 3, 4, 5, and 6. We took actually 10 sequences randomly. And it was also because this was the first time, so we did not know what will work. So we took sequences in one direction, in another direction. We took long sequences, shorter sequences, overlapping sequence, non-overlapping, and we were hoping that something will work, and we wanted to make sure that this is the first experiments of its kind, and we make sure that we put all the controls. The odds against this idea were many. This was the first such attempt, and all the sequences that we considered, they did not resemble any known gene. They were not conserved evolutionary, so somebody would say that, okay, if they're conserved, that means they must be doing something in the organism. So we took non-conserved sequences. And GenMark, one of the best tools at that time, said that they are not ORFs. When I presented this work to students, they all refused to work on this project because they said there's no previous publication. So If they fail after 1 year or 2 years, then they'll be in trouble because who will take the responsibility? So anyway, we respect their decision. And so we hired, and this work was done in Japan. We hired Chosu from Singapore and as a technician. And she worked in the lab, and she had to be trained in molecular biology, first of all. But after that, within 6 months, she worked in this project and she got results, and she was co-author on a paper. As you know, having a technician as a co-author is really something significant, but we respected her contribution. Well, the question is, how do you make a gene out of non-expressing DNA, which does not make proteins or RNA? That's pretty simple problem, because you put the non-expressing DNA sequence, and the textbook says that if you put certain things upstream and some signals downstream, then you have eventually created a functional gene out of it. But is it possible? What is it scalable? And what are the constraints one has to come across? So if you play with the genome sequence, you know very well that this genome sequence, you're trying to install some sequences, some markers, and then you have to make sure that these markers stay there. And also, they don't mutate. They are in a fixed order. Now, if you have to make one functional gene, then it will be very difficult to maintain the quality and takes a lot of time. So we said, let us not even get to the genome level. Let's pull this information out of the cell and make genes outside the genome. What this means is, so we were looking for an expression vector, and then we were fortunate that in vitro gen had released this PBAT topo at that time. And this PBAT topo gave us epitope, it gave us start ribosome binding site promoter sequence upstream and a stop sequence downstream. I'm sorry, I'm getting some background. So is it possible to mute? Hello. Is it possible to mute? Is it possible to mute? Yeah. I'm getting background, please. Yeah, okay. Thank you. So we got this in vitro gen vector, and we amplified the sequences. So whatever was bioinformatically approved, we took those sequences and we amplified them. We inserted them in the PBAT topo, the place where you see the yellow colored region. And now, suppose your protein resembles 100 other proteins in molecular weight. How would you make sure that it is your protein? So we were able to use this epitope, the his tag, and the antibodies are already raised against that. They're available in the market. So all we have to do is to express the sequence and to do the Western blot. And if there is a band that would show that the protein is made, that is the one, it is made from intergenic regions. If there is no band, it would mean that there is no protein. And finally, the defining moment came when we got the proteins in presence of the inducer. We got the band. In absence of the inducer, there was no band. By indicating a linear correlation between the induction of this particular expression, inducing this expression, and the stability of this expression. So this was the first time we were able to get the functional proteins from intergenic sequences of E. coli. Okay. And the next question, now the nature of questions changed. And the next question was, do these polypeptides or proteins fold into a certain structure? We did some computational analysis quickly, and then we found that, yes, out of 10 sequences that we initially took, we lost 4 of them, and 6 survived. And then out of 6, we found 2 of them have some structural correlates which are very attractive. Now the point is that what do these genes do in the cells? What are they doing? How are they impacting the cell? We wanted to catch the phenotype of the transformed cell. And so we found that when we express these genes in the cell, all the cells looked similar in their shape. And then when we checked the cell growth, we found that out of 6 gene expression events in different E. coli cells, one of the E. coli is declining in terms of numbers after 2 hours of expression. And so we said that if it is really killing the cell, if it is really the authentic observation, then by switching off the gene expression after 2 hours should restore the growth. So that is the question we asked. And that's exactly what we found, that after 2 hours, we are able to restore the growth back up again when we remove the inducer from the medium, and we cultured the cells in a medium that does not have inducer. And we found that the cell growth is restored, thereby proving a linear correlation between the expression of the gene made from intergenic sequence and the phenotypic outcome. The functional study of the lab genes can only be made. So far, we are looking at this evidence. They may be made, functional study of these genes may be possible if we overexpress them. Now, we do not know if you delete intergenic sequence or you delete a not coding sequence in a desert, in a gene desert, in the genome, what will happen to the cell? Because all the knockout library that has been made with reference to the protein coding genes, which are actively there in terms of their natural expression. But what would happen if you delete a part of the intergenic sequence? Will the cell sense it, or will it ignore because it never used it anyway? We do not know at the moment. But what we know is that if you overexpress the cells, overexpress the genes in the cells, then something may be perturbed and something may be disturbed, and we can catch this disturbance in the form of the microarray data analysis and then design suitable phenotype assay. So the ENCODE project has established that a tiny part is proteome, bulk is transcriptome, and some of it is not coding. So now we are, our assumption is that we can probably convert the transcriptome into proteome, but we are definitely going to come across a large number of failures. That large number of failures will be a new knowledge anyway, and that will also tell us something interesting about biology. So the question is, how many intergenic sequences can be converted into brand new genes? Or, in other words, how much of transcriptome can be converted into proteome, right? Because now you have a technology that can allow you to express the sequence of your choice. So this led to Encyclopedia of Lab-Born Gene project, and it started from my entry to Kerala, University of Kerala, where we started the Center for Systems and Synthetic Biology at Trivandrum. And then from there, I moved on. After close to 2 years, I moved to Pune and now to Delhi. And so this work has been going on, and at the computational level. So the idea was to profile intergenic sequences, introns, pseudogenes, and so on for making novel proteins and peptides. Can we make a functional protein from an intron? Can we make a functional protein from a repetitive DNA sequence? And there are other, origins, and I'm coming to that in a moment. So this led to a new drug discovery platform, because we were looking at functional correlates at the therapeutic level. And as a result of that, we found a new drug discovery platform. So I'm going to give you a very quick snapshot of the results that we got. So we found a yeast, this came from Drosophila, and we found that a sequence that was an intergenic sequence in Drosophila, when we chemically synthesize the peptide of this sequence after a lot of bioinformatics filters, there are a large number of bioinformatics filters, and finally out of hundreds of sequences, we zoomed onto some sequence which was showing a structural similarity. That means the DNA sequence, when it was computationally expressed into a protein sequence equivalent, and then we did a structure blast, which means you do not match the sequence, you match the structures. And the structure blast indicated that close to 99% of the structure was similar. Sequences are different, as you can see, yellow and blue sequences. These are protein sequences. They are different, but the structures are nearly similar, nearly identical. So therefore, we said that can we synthesize this peptide, which is coming from where? The Drosophila intergenic sequence. Can we synthesize the peptide and find the function? And we found that it kills E. coli, kills Staphylococcus aureus and other organisms. And this was very interesting because our reference point was a defense molecule. The yellow portion that you see is a commercially available defensin, alpha defensin molecule. And we found that when we convert an intergenic sequence into an amino acid sequence equivalent, and we are getting nearly identical structures, a computational prediction is leading to an experimental outcome which is very similar to what has been predicted. Likewise, we received some more peptides. This was from yeast intergenic sequence. So we collected nearly 2,000 intergenic sequences. And from there, we did the structural and sequential similarities. And we matched them against, this was a target-based study, wherein we chose 3 malarial targets, and EBA 175, MA1 and MSP 119. And we raised the peptide against these targets, and then we did some docking studies. And the outcome of that docking studies, of a large number of docking studies, we finally chose 3 molecules which were showing strong correlation. I would again like to emphasize these peptides were coming from the intergenic sequences of yeast, Saccharomyces cerevisiae. And when we chemically synthesized them and tested them on the malarial parasites, we found that a large number of malarial parasites are unable to enter RBC in the presence of these peptides. So this was another encouraging response. And then likewise, we found a novel peptide that kills leishmania. And this is a very recent story. So we asked that tRNA never got a chance to translate itself. In evolution, it is only doing some social outreach program by carrying on the amino acids to the ribosome level, helping the DNA to translate and into a certain polypeptide. So it has always been doing this kind of, the service model for a large number of years, maybe millions of years or billions of years. And so tRNA never got a chance to translate its own sequence because of the steric hindrance and maybe other reasons. So how about if we translate tRNA into a peptide sequence equivalent and see what happens? Of course, we cannot do this in vivo conditions. So we took a large number of tRNA molecules, and then we bioinformatically determined a lot of properties, and then put some filters, and finally we chose one molecule which seemed to be very interesting. And then when we chemically synthesized this tRNA peptide, and we tested it against a lot of cell lines and microbial screens and all that, we finally found that it has an antiparasitic property. In leishmania, it is able to, and you can see the electron microscope pictures wherein the cell in presence of this tRNA encoded peptide is swelling and it gets ruptured and it kills the cell. Likewise, we have found anti-breast cancer molecules which are enhancing the effect of the anti-cancer drug. And we have found some novel candidate recently for the Alzheimer's also. And this is all the work in progress, and we are filing the patents of these molecules very soon. So like that drug repurposing, well, it's anyway old story. This is nothing new from our lab. This has led to Foresight Biotech Private Limited. It's a 3-year-old startup focused on R&D and recognized recognized by the government of India. And so we said that now we have got the proof of the concept. It's time to scale up. And so we have this company, and we are getting funding from investors and other people to scale up and bring the product to the market. So if you're interested in this, please feel free to write to me. So we, so this we published the paper back in 2009, and 2010 I was invited to European Science Commission to present this paper in Brussels, and they were very generous to give us good reviews. And likewise, after that, U.S. National Academy of Sciences at Beijing. This was a meeting with the Beijing Academy of Sciences, and then the IUPAC, IUPAC, and the other. What you see the building here is an organization for prohibition of chemical weapons. So I was invited for giving talks at all the places. And one of the interesting thing happened here at OPCW, that somebody asked that since we are able to make a lot of genes from intergenic sequences, and can we also, does this have a potential to make bioweapons? We didn't think about it before because this is not what we were, set out for. But then this suddenly came as a realization that it is possible for people to, misuse the technology. So we are very careful not to make anything that looks nasty. We have, focused ourselves on the therapeutic domain, and we would like to continue offering new drug discovery approaches and a new drug discovery platform. So currently we have a 500 molecule inventory, which is growing every day. And the best part is that you own the IP of every molecule that you make, because this molecule was not made by nature. So when you make the molecule, you own the IP, intellectual property. Everything started from junk DNA, and then it became junk gene, junk DNA, RNA, and proteins, and all that. So we, my students sometimes like to call this area as junkomics, as a new area of making functional components from these sequences. We have been offering this training program for the last 3 years. And now we are at the last batch. It will be difficult for people to, because I think more or less the selections have been made. So this is a collaboration between 3 organizations, JNU, CUSAT, and Pondicherry University, where we offer a very detailed 6 months training in synthetic biology. And this has been going on and quite successful. The first event that we organized was in Kerala. This was Biodesign India, wherein we created this community of synthetic biology people when I came from Japan. That was the time when we started this. And then we have been organizing Indo-US synthetic biology events, and also some events in JNU. To expose the whole field to the community. And I'm so delighted that MKU is now taking a lead and recognize the importance of this. So we can all work together in future in case there are more opportunities. So there are, of course, challenges always, both in terms of science and in terms of spreading the information, distributing this information, and also generating new ideas and bringing them to the industry level. There are challenges at every level, but we receive motivation from an atom that says, be positive, even if you're always surrounded by negative things. So thank you very much for your kind attention. It was lovely talking to you.